Hello, my name is Brandon Gamlin, and along with my partner Kyla Hutzel, we will discuss in this tutorial video the dark reactions of the Calvin cycle in plants. This reaction occurs in the stroma of the chloroplast with the intent of fixing atmospheric carbon dioxide into carbohydrates, specifically hexose sugars. As with all cycles, regeneration takes place, this being the acceptor molecule for the carbon dioxide. The light reactions have set up a precursor to this reaction and supply the ATP and NADPH needed. The Calvin cycle is done in two stages. Beginning with stage 1, carbon dioxide is fixed, trapped as a carboxylate and reduced to carbonyl level in sugars. This is where glyceraldehyde 3-phosphate, otherwise known as GAP, is produced. This GAP will be later used to make hexose phosphates and polysaccharides. Going into stage 2, GAP is used to regenerate ribulose 1,5-bisphosphate, otherwise known as RUBP, which is the acceptor molecule for carbon dioxide. Carbon dioxide is first diffused into the stroma of the chloroplast, where it is picked up by the carbonyl carbon of the acceptor molecule RUBP. The catalyzing enzyme for this is ribulose 1,5-bisphosphate carboxylase, otherwise known as Rubisco. The carboxylase reaction is complex with many steps as shown here. In step 1, the Rubisco enzyme is activated by carbamation of a lysine residue's active site and deprotonates a carbon on the RUBP, which forms the enediolate intermediate. In step 2, the enediolate intermediate is carboxylated to form a carboxy-beta-keto intermediate. In step 3, the previous carbamate from step 1 forms part of the binding site for a magnesium +2 ion. This ion is then included in the binding of the RUBP and activating a water molecule which hydrates the carboxy-beta-keto intermediate. Step 4, the hydrated intermediate is then cleaved by the protonation of an active site acid. The cleaving produces two molecules of 3-phosphoglycerate, which is also known as 3-PG. These steps leave carbon dioxide fixed into a carbohydrate with production of the hexose and regeneration of the RUBP remaining. The next chemical reactions involve the previously made 3-PG. Starting off the reaction with step 1, each of the 3-PG molecules are phosphorylated by ATP, which is catalyzed by the enzyme phosphoglycerate kinase. This produces two 1,3-bisphosphoglycerates. Step 2. The 1,3-bisphosphoglycerates are then reduced by NADPH and catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase to form two glyceraldehyde 3-phosphates. At this point in the cycle, two 3-carbon monosaccharides have been produced, and for each carbon dioxide molecule, two ATP and two NADPH have been used. This means that 6 molecules of carbon dioxides have to enter to provide the 6 carbons needed to form each hexose produced. In total, the formation of 12 GAP and 12 ADP and 12 NADPH are needed. A split in the pathway happens as the two goals of the cycles are being addressed. First, we will discuss how the formation of hexose sugars proceeds, and then the regeneration of the acceptor. Of the 12 GAP formed, 2 will be used to make a molecule of a hexose. In step 1, a molecule of GAP and DHAP are combined with the help of the fructose bisphosphate aldolase enzyme to produce fructose 1,6-bisphosphate, otherwise known as FBP. In step 2, the FBP is then dephosphorylated by fructose 1,6-biphosphatase, which in turn yields 3 molecules of fructose 6-phosphate, F6P. In step 3, the F6P is then isomerized to yield glucose 1-phosphate, G1P. Glucose 1-phosphate is then taken up by oligosaccharide and polysaccharide procedure to form sugars such as glucose and sucrose. Next is the regeneration of the acceptor. The remaining 10 GAP, 2 DHAP, and 2 F6P from before will now be utilized to regenerate the 6 molecules of ribulose biphosphate needed to keep the cycle going. These molecules undergo transketolases and transaldolases to rearrange and recombine in this procedure to produce 6-ribulose 5-phosphate. In the final step of regeneration, ribulose 5-phosphate undergoes phosphorylation catalyzed by the enzyme ribulose 5-phosphate kinase with the inclusion of ATP to form ribulose 1,5-bisphosphate to complete the regeneration. In summation, with the requirements for synthesizing the 1 mole of hexose from 6 molecules of carbon dioxide, 12 moles of NADPH, and 18 moles of ATP, the overall dark reaction can be written as follows. Thank you, and we hope our video was helpful in understanding the dark reaction of the Calvin cycle.